Hello students, in today's session we will be learning about chromosomes, chromosomal theory of inheritance, which is covered in your chapter Inheritance and Variations. So let's get started. Students, Mendel published his work on inheritance of traits in 1866, but for some reasons it remained unnoticed or unrecognized till 1900. As Mendel's was much ahead of his time, the scientific community at that time failed to understand the depth of his work. In 1900, three scientists, Hugh de Vance, Corin, and von Schirmack, independently rediscovered Mendel's work on inheritance of traits. Due to advancement in microscopy, scientists were able to observe cell division and the structure of chromosomes under the microscope. At that time, we had two scientists, Walter Certain along with Theodore Bouvier in 1903, who studied the parallel behavior of Mendel's factors, that is genes, and behavior of chromosomes at the time of meiosis. Based on these observations, the chromosomal theory of inheritance was put forth by Certain and Bouvier. Students, let us see what this chromosomal theory of inheritance states. The theory states that the chromosomes are present in pairs in a somatic cell. So you need to have a pair of a chromosome. During gamete formation, homologous chromosomes pair, segregate, and sort independently during meiosis. Thus, each gamete contains only one chromosome from the pair. Nucleus of the gametes contains chromosomes which carry all the hereditary traits. Male and female gametes, that is the sperm and the egg, carry all the hereditary traits. They are the link between parents and the offsprings. The fusion of the haploid male gamete and the haploid female gamete restores the diploid number of chromosomes in a species. Students, to understand the chromosomal theory of inheritance, we need to first understand what is a chromosome. Chromosomes are filamentous bodies present in the eukaryotic nucleus. This term chromosome was first coined by W. Wilder. The size of the chromosome varies from species to species. Each metaphase chromosome varies from 0.1 to 33 mm in length and 0.2 to 2 mm in thickness. Now, chromosomes are visible only during cell division. They are capable of self-replication and they play a very important role in heredity, mutations, variations, and evolutionary development of eukaryotic species. Chemically, eukaryotic chromosomes are made up of DNA, histone, and the non-histone proteins. Now, to understand this, I have just put up a picture here. See, there is a cell. This is the nucleus of the cell. Normally, you have the genetic material that is just suspended in the nucleus. But at the time of meiosis, you have these chromosomes, the genetic material that gets arranged in the form of the chromosome. I've just picked up one chromosome and highlighted that for you. Just have a look at this chromosome. You can see how it is then broken down to its individual components. You get your DNA strand which is actually wound to its nucleosomes, and you have these proteins that along with the DNA, you have the histone proteins, you have certain additional proteins which help in arrangement of this DNA. Those are called as the non-histone proteins, and you reach out to the individual double-stranded DNA. Now, genes are located here on your DNA, which are responsible for the inheritance of the trait. The most important point to remember here is you can see the structure of chromosome only at the time of cell division. The function of a chromosome: chromosomes are carriers of heredity. Number of chromosomes. The number of chromosome is specific and is constant for a particular species. Therefore, it is of great importance in the study of phylogeny and taxonomy of a species. The term ploidy speaks for the degree of repetition of a primary basic number of chromosomes, that is X in a cell. To understand this better, the primary basic number of chromosomes in a human cell is 23. So that is 23X. And in the diploid state, it is 2X or 2N, where it becomes 46. When the chromosome number in a cell is the exact multiple of the primary basic number, then it is called as euploidy. So basically your complete set of chromosome that is present in a cell is an example of euploidy. Euploidy includes monoploidy or the haploid, where you have only one set of your chromosomes, where X is just N. Diploid, it is 2N. I said single 23 in humans, and diploid it will be 46. Or say two sets, 23 pairs of chromosomes. You also have triploid conditions where it's 3N, tetraploid condition where it is 4N, and so on. Now, when the chromosome number is not the exact multiple of the haploid set, then it is described as aneuploidy. So aneuploidy is basically either an addition or a deletion of one or more chromosome to the total number of chromosomes in a cell. Now, why do we study aneuploidy? Because any addition or deletion of the number of chromosomes may lead to genetic defects. Here, this particular chart shows the variations in the chromosomes. You can pause the video here and have a look at this chart. Let's have a look at the structure of chromosomes. Chromosomes, as I've said, are best visible at the time of metaphase because in this stage they become highly condensed. Now, this is how a typical chromosome looks. It has two chromatids and which are joined together at a place called as a primary constriction or a centromere. The primary constriction consists of a disc-shaped plate called as, this is that disc-shaped plate called as kinetochore. Now, what is the function of this kinetochore? You have the spindle fibers which come and attach here at the time of cell division so that the chromosomes can be broken at this point when the cell is dividing. Besides the primary constrictions, some few more constrictions are present in a chromosome. Here, I've taken the single strand to explain the primary constrictions. You can see the primary constriction 1, secondary constriction here 2. Now, these all are required for the organization of the nucleolus. You have a small satellite body which is attached at the secondary constrictions in a few chromosomes. Now, each chromatid in turn has a long, unbranched, slender, highly coiled DNA thread called as chromonemata extending through the length of the chromatid. So, I have taken this individual chromatid here and you can see here right from the start to end you have the double-stranded DNA that is present. End to end of your chromosome. Types of the chromosomes, depending upon the position of the centromere, there are 4 types or shapes of chromosomes. Have a look at the picture here. When you have the centromere exactly at the center, it is called as a metacentric chromosome. When it is just a little above the center, it is called as the submetacentric chromosome. When it is towards one side of the chromatids, it is called as an acrocentric chromosome, and when it is completely at one end, at the tip of your chromatids, it is called as a telocentric chromosomes, and they have different shapes. When they appear, they show different shapes under the microscope. Students, the next thing that we are seeing is sex chromosomes. Now, the chromosomes which are responsible for the determination of sex of an individual are known as sex chromosomes. Allosomes. Human beings and other mammals have X and Y chromosome as sex chromosomes. Here, I have shown you a diagram of the structure of your X and Y chromosomes. If you closely see, you will see that the X chromosome is quite bigger in size as compared to the Y chromosomes. Now, both these chromosomes have 2 regions. One is the homologous regions where you have the same type of genes, and the other 2 is the non-homologous regions which has different genes. Now, the non-homologous region of your X chromosome have X-linked genes, and the non-homologous regions of your Y chromosome has Y-linked genes. Normally, crossing over occurs here in the homologous regions, but these genes that are present here, which are called as X-linked genes, these are called as hemizygous genes. Here, these are also called as hemizygous genes. That is, a single pair of gene is sufficient to determine the trait. I have also put here a distinguish between your X and Y chromosomes. Now, for the female determination of sex, you require two X chromosomes, and for the male, you require one X and one Y. So, you have this X chromosome is a sex chromosome that occurs in pair in female and single in male, whereas you have Y chromosome which is only present in the male. Now, this particular X chromosome is required for female sex determination and Y chromosome for the male sex determination. It is much bigger in size as compared to Y chromosome. The diagram itself is explaining this particular point. Represents 5% of the entire human genome. It represents only 2% of the entire genome. This contains more genes. This contains fewer genes. It is obvious from the diagram. Contains the XX genotype. This contains the XY genotype. Now, so this is your genes. Now, to understand these X and Y chromosomes, this diagram explains it all. So here you have similar genes and here you have your dissimilar genes. So this is the non-homologous region. This is the homologous region. This non-homologous region has X-linked genes of your X chromosome, and this non-homologous region of your Y chromosome has the Y-linked genes. So what is X-linked and Y-linked will be explained to you further. Students, in the previous slide, I already explained the X and Y chromosomes, how they interact with one another. Crossing over in the X and Y chromosome takes place only in the homologous regions. The non-homologous region of the X is longer, it contains more genes, and the non-homologous region of Y is comparatively shorter. The Y-linked genes that are present on the non-homologous regions of X chromosomes and X-linked genes are present on the non-homologous region of X chromosome, and Y-linked genes are present on the non-homologous regions of the Y chromosomes. Now, when you're talking about interaction, there is a step called as crossing over in your meiosis. Now, this occurs in prophase 1 when parts of homologous chromosomes overlap. Here, you have your two homologous chromosomes coming together, and there is a formation of a synapse when they come together, and when they switch their genetic information, that particular stage is called as a crossing over. Students, crossing over is a process that produces new combinations, which is also called as recombinations of genes by interacting and exchanging of corresponding segments between non-sister chromatids of the homologous chromosomes. Now here, these two are called as sister chromatids, and the cream color and the green color segments are called as non-sister chromatids. Now this occurs, I told you, in the stage of meiosis, the Pachythin stage of prophase 1 of meiosis. Now, this term of crossing over was coined by Morgan. The mechanism of crossing over consists of 4 sequential steps, such as synapse, that is, they come together, tetrad formation. You can see all are together, 4 chromatids. So that's your tetrad formation. Then you cross over, you exchange the material here. And then there is terminalization. Once the exchange is taking place, the individual chromatids separate out by exchanging the genetic information. The phenomenon of crossing over is universal because it increases chances of variation. Here with this picture, you can clearly see how the chromosomes come together, cross over, exchange the material, and then independently separate out at the time of gamete formation. See, note here, when the gametes are formed, this particular gamete, the first one and the last one, shows the parental combination, but the middle two gametes show you the new genetic makeup, which is a new gene combination. This phenomenon, students, is called as genetic recombination. Thank you, students, for participating in the lecture. We will meet soon again. Till then, take care, be safe, and keep learning.